can already start by welcoming everybody, and still people coming into the virtual room and people still coming into the actual room. So welcome everybody. Thanks for joining us for our monthly West Coast Metabolomics Center seminar series, as well as supported by the UC Davis Genome Center, the focus group Data Science and Metabolism. So thank you so much for joining us. And with this support, we are able to get people in. And today I am very happy and glad to welcome Professor Jan Krumsiek, if I pronounce it correctly. Correct. You know. Well, sometimes people choose to have a different pronunciation after a while, right? Professor Krumsiek is Associate Professor of Computational Genomics at Weill Cornell Medicine in New York. He was before an assistant professor 2018 to 2024. And he got his Master's of Science in Bioinformatics in 2009 and the PhD in 2012 from the Technical University of Munich in Germany. After that, he became a postdoc, 2012 to 2016. He was postdoc and then a team leader, junior group leader, which is kind of like an assistant professor to some extent, at the Helmholtz Center in Munich from 2016 to 2018 before he then went to Weill Cornell. He develops and applies novel data analysis methods for metabolomics and multi-omics data sets, pathway-based methods, network approaches, machine learning techniques, and deep learning method models, specifically metabolic data sets in the field of diabetes, cancer research, and Alzheimer's, and of course also published extensively on the topics of etiology and risk prediction. Now with that is great, but he also said something interesting, which I found online. He said, a lecture where we spend half of the time on questions that came up from the audience, that is a great lecture. So that means don't feel shy, right? It means like you have a question, a burning one, say so. And you have another question that is less burning, keep it to the end, but don't forget, a good lecture is only a lecture when a lot of questions are there, both online. So we have people who look at online, and online you can put it into the chat room. So we monitor the chat room at all times. Two people actually monitor the chat room.
And I'm seeing it too.
And the speaker is seeing it too, yeah. Right? So three people that monitor it. So don't be shy. Put things on it, in the chat while the speaker is speaking. So then they can also relate back later after the lecture. Okay. So then we can talk about those all together. And otherwise, yeah, the floor is yours. Thank you, Jan, for coming.
Thank you, Oliver, for the nice introduction. Hi, everyone. Hi on Zoom. Sorry to the Zoom people, you'll see this camera and I might be walking in and out of it, but I think the slides are more important anyway. So today I'm going to cover a bunch of topics, sort of an overview across different fields. The style of the lecture is more flying over the topics. I want to rather show you all the things we've done rather than go into one computational methods in detail. I'm going to mention a few things for the computationally inclined here, but it's more about presenting the findings. If you have more questions, then we can talk after or another time also, if you really want to know the nitty-gritty details of the computation methods. I'm going to go until like 50 minutes in, so we have time for questions after, or if people have questions in between, please ask. If I don't make it through, I can jump. The only thing I will definitely show is this last one because that's the newest, so whatever happens, I'm going to jump there. So with that, I'm going to start the journey with some of the old stuff. So for me, metabolomics was pretty new about 15, 16 years ago. I guess it wasn't that new maybe, but still in the early phases. And in the beginning, we had some, I think, interesting findings that were not even related yet to diseases as such, so predicting disease or understanding disease, but also just what are these metabolic measures themselves and how are they internally structured. So that was the field of the early metabolic networks, as I said, not even necessarily disease related. So one of the findings that I found was fascinating from back then in my PhD, so this is some old stuff, is that when you do these correlation networks, so these were blood samples, about 2,000 blood samples from the CORA cohort. Back then we had about 517 metabolites measured. This is this Metabolon platform, so about a third or so is still unidentified features, all the white bubbles. And then when you make these metabolic correlation networks, in this case a special measure, partial correlation, kind of deconfounded correlation, what you see is sort of pathways emerging, and not just sort of pretty pictures. The one on the left side always looks nice, but if you really look into sort of the individual cascades, how the statistical model, that knows nothing about biology, of course, correlates the measurements of these blood markers, you can see that there's actually individual biochemical steps acting, right? So specific pathway here, you can see the amino group going off from here to here, no directions, because statistical models are very bad at causal directions. Or from here to here, you can see that sugar residue changing. So what was interesting, and that went through different papers, it's so accurate, even though that it's in blood, that you can reconstruct sometimes reaction by reaction what the biochemistry looks like just by putting in the data. And to the point that in this paper, the big story was you can actually get a prediction of the unknown metabolites, of your unidentified peaks, right? Because you have something like known, known, unknown, known, known, you might actually figure out from biochemistry what it is. And sort of maybe identifying unknown metabolites using statistical networks is not everyone's interest. But what I thought was the interesting part is that even though this is blood as a medium, a couple membranes away from where the actual chemistry happens, you can still see those pathways represented and able to be picked up by statistical methods. That was my fascination for my PhD, and maybe also good news for blood biomarkers and making sense of all these metabolites we measure in blood, because it's not just some random molecule that ends up in blood, but it still carries a deep imprint of the cellular dynamics in it. We later also did this with genotypes. I'm going to skip over this real quick. And then also, this is now we're jumping 10 years ahead, or almost 15 years ahead. Another version of sort of the same message was we used variational autoencoder. You can call it deep learning. I think deep learning is poorly defined these days. Someone once said that if in these networks you have 2 layers, that's deep learning. If you have only 1, it's not deep learning. But anyway, in this new realm of sort of the network-based methods, what a variational autoencoder basically is you put your data in, you force it through a bottleneck of compression, and then you try to reconstruct the same data. The idea being, if I force information through a bottleneck, what's left is what actually matters, right? So a little bit like data compression. If everyone remembers MP3 files for audio, they get rid of the stuff that doesn't matter, and then you reconstruct almost the perfect sound. So it's kind of like that, but you don't compress it to store it on a hard drive. You compress it to remove redundant information. That's the idea of a variational autoencoder. Actually kind of similar to principal component analysis, by the way. And so what we did here is we fitted one of those variational autoencoders on the Twins UK, also Metabolon data, heavily skewed female cohort, and sort of then you can analyze these latent features. There's all kinds of pathways in there. But what the fascinating finding was, if you now take these latent dimensions, so these lower level representations of the data, which are composed each of multiple metabolites, and look for a disease signal in completely different cohorts, you find very interesting associations. So specifically, we looked at the QM diet cohort, totally different. It's an Arabian cohort with untreated diabetics, so totally different than the original Twins cohort, right? We looked at a leukemia study, that's an American study, and a schizophrenia study, again an Arabian study, and we projected what we learned in the UK data and onto these disease data sets. And basically what you see is, sorry, I know this is a lot of information, but basically listen to what I'm saying instead of looking at too much, is these new latent dimensions, sort of those inner workings of metabolism, how it's composed on the inside, has predictive value on diseases in these completely different cohorts. So that's why we said you learn transferable representations of metabolomics data. So one more time, you learn an inner feature of metabolic wiring on highly female skewed British, mostly Caucasian white data, and look at that in completely different cohorts and find high disease correlations, in fact, higher than when you do principal component analysis or other models. So that was sort of another version of this, that there's some inner wiring of blood metabolomics that seems to preserve the pathways that originally acted on these metabolites in some cell in the body, and they seem to be pretty conserved across different populations, genders, ethnicities, and so on.
What about species?
Species. So we have a project. So if you talk about mice, that's difficult. So we know, and I'm going to talk about that for a minute later, is that, for example, blood metabolomics profiles in dog lymphoma. Sorry, I don't know who asked the question. Sorry, I was just looking in that general direction. The blood metabolomics profiles are in dogs happen to allow for a similar stratification of patients as in humans. But that is only because they have the same subtypes of cancer, right? So if the biology itself were different across the species, that wouldn't work. The advantage with the species, of course, is, and this is something good for the metabolomicists and tough for the genomicists, is that there's no such, you don't have a dog glucose, you have glucose, right? So the molecules are the same, you don't have to do homology mapping or anything. But I would say it depends. So if the underlying biology and disease is not the same, then the blood signal also wouldn't be the same.
I was wondering how big was the input dimension to the original input?
So we had 217 metabolites. That was the input. This is why I'm careful saying deep learning, because it's not so many parameters as many of these modern models have. 217 shared, because that was the only ones that were shared across all data sets. And then the next latent dimension was something like 20, 30, and then down to a smaller bottleneck. Yeah. So that was sort of a bit of a blood metabolomics and its general features. Now we're actually going to go into some of the disease studies that we've been doing. Alzheimer's disease nutrition is sort of related to some diseases, and then cancer. And again, the last one I definitely want to get to because that's the newest information, the newest project. So in Alzheimer's disease, probably everyone knows about it. So well, I almost said driven, I had to catch myself. It is associated with amyloid beta built up in the brain, and tau pathology, causality heavily debated. Some drugs are on the market against the amyloid protein, also heavily debated. One was retracted recently. Frustrating disease, probably starts way earlier than the symptoms present themselves. And still very rich field of research, and you can look at it from a central metabolism, so the actual brain, if you have access to it, or peripheral metabolism, blood samples. So I'm going to first show you one of the brain studies that we have, and here's a big disclaimer for everything that I'm going to show you in the next 5-6 slides or so is, this is post-mortem brain. And it has to be because in the human system it's the only way we can access those brains. There are very, very rare studies where you can have actual live brain tissue from surgery leftovers. But usually brain biopsy in a healthy individual is not really something that's happening, right? So we have to rely on post-mortem brain in these carefully collected studies. But you have to know that, of course, they underwent a catastrophic event, death, right? And then that's the tissue you're working on. But it's the best we have, so that's what we're analyzing. So these are dorsolateral prefrontal cortex post-mortem tissues from the ROS-MAP cohort. The nice thing about the ROS-MAP cohort is these people also have been in the study for many years before death, so we have longitudinal data up to the time point. And then after autopsy, you have brain pathology with all these measures of how sick they were in terms of Alzheimer's disease, and metabolomics measurements. So just a bit of a general overview. I know it's always like a lot of data on these slides, but so you've the composition. This is not the metabolic composition of the brain. This is the metabolic composition of the platform we're using, which is again the metabolome platform. So if this says 42.7% lipids, doesn't mean all chemicals in the brain are 42% lipids. It's the platform skews heavily towards lipids. So this is just what's detectable in these brain samples. And then you have all these different measures, hope you can read it in the back, of pathology that in one way or another tell us how sick the brain was in terms of Alzheimer's, or how their cognition was affected before death, because we knew these people, right, before they passed away. And just because it's a lot of data, you could go through this for a long time, because you associate all the metabolites statistically with all those outcomes, you have to put it together somehow. So I'm just going to give you some. This is one of our highlight slides, but you could go in the data and search forever. So what do you see in a sick brain? So you see upregulation, I should say, higher concentrations of cholesterol metabolism, which also goes in line with Alzheimer's being known for having a strong cholesterol metabolism component. That's the ApoE gene, actually, is the main genetic predictor of Alzheimer's disease. We see a deregulation of bioenergetic pathways and mitochondrial pathways. I'm going to come back to that later. You see sort of inflammation up, anti-inflammatory signals down. That makes sense. And then this is technically not a pathway, but more sort of a general group of osmoregulation, so metabolites that have to do with the water pressure and osmotic regulation in the brain, which actually has a strong connection to cognitive decline as well. That one is particularly speculative. It's the observation you have if there's actually a direct sign of osmoregulatory changes in the brain that have to still be determined. That's sort of the general picture of the main changes, metabolic changes we see in these brains. And then we also addressed a particularly hot side question, and I'm not going to give the general answer, but our answer to this is amyloid and tau, right? So in the as I kind of alluded to, there's this big debate in the field. Amyloid are those folded protein plaques, and whether those are the causal protein or just a side effect of the disease, right? And if they're not the causal protein, then all these anti-amyloid therapies that everyone's talking about are simply not going to cure the disease. And tau is that other protein that's inside of the neurons. It sort of shreds them apart. That has a lot of attention, but it's also not clear, is it secondary, is it primary? And very hotly debated. Every year at the Alzheimer's conference, there's like a sticker poster where people can say what they believe these days. And so the true answer to this question will matter, but at least what I can say, what we found is there was some statistical trickery. So you look at the amyloid effects correcting for tau, and then you look at the tau effects correcting for amyloid, is that tau has an independent signal with respect to its correlation with metabolomics, but amyloid does not. So first of all, tau has more hits. There's more metabolites correlating with tau. And even if you correct for amyloid, most of them survive. If amyloid has less hits to start with, and if you account for tau, they're almost all explained away. So there's no independent amyloid metabolite correlation, at least in the data that we have. And we repeated the same experiment. There's also a matching proteomic data set for the same brains, and it's the same effect. So in a nutshell, again, I'm not saying which one is the causal, I don't know, but at least the statistical correlations with metabolites are tau-driven in this data set. They're not amyloid-driven. That was our 2 cents on that messaging. Again, causality is a different beast. And then I'm going to, because for the sake of time, there were sub-variations of this, what we did in the future. So we looked at it also in ethno-racial diverse groups. So the question basically being, the signal is very subtle, very, very subtle. So what you're supposed to see here, and statistically it's significant, but it's hard to see, is that in the African Americans and the Hispanic subpopulation, you have a sort of in these metabolites, in these sphingometabolite, no association with Bragg stage, whereas in the whites it goes down, and so it's a small subtle finding. It's a very subtle finding. So this idea that even the metabolic dysregulation itself is now different across ethno-racial groups. This will be a very small paper because that's the end of the story. That's all I can tell you. I don't know why, but we see it. I'm going to skip over this part. So that was the brain metabolomics, and there's still more of these post-mortem samples coming in everywhere. And again, it's always post-mortem. If you want fresh fresh brain, you would have to do it.
Which
in
region
animals.
was it?
Sorry, yes. So this was the dorsolateral prefrontal cortex. And then we had another, maybe I'm going to show this real quick. We had another study with Alzheimer's samples and PSP samples, palsy samples. And so that was in temporal cortex and cerebellum. And the interesting finding in that particular study was that the cerebellum, at least from literature, is thought to be mostly disease-free. No, not disease-free, but not as affected in terms of how the disease goes through the brains temporally. In the end, before death, everything's affected in the brain, but it's not thought to be one of the first regions affected. But metabolically, we actually did see a lot of effects in it, whereas in the temporal cortex, which is thought to be affected early on in Alzheimer's disease, we did not see a lot of metabolic effects.
I was just wondering if you've had a chance to take this data and look at the longitudinal measures of white matter hyperactivity, because you do have those in process.
Yeah, so we are currently, in fact, so that's now the upcoming projects where we're trying to connect a little more so because here we did use the at lifetime data, but only the only longitudinal aspect we really used was the trajectory of memory. So if they are declining fast or slow, that was it. And now studies are coming where we also have the blood samples of the longitudinal, sometimes even multiples, and then the accompanying longitudinal features and all of the imaging and so on. And that's only upcoming now. Yeah, but yes, it's part of the plan. Yeah.
Question from the chat: Where are you able to look at genetic ancestry in analysis, including Hispanics? How levels vary depending on ancestry proportions of individuals in that group?
Yeah, that's a complex question. I think the biggest challenge in setting up this ethno-racial study was the definition of those groups, right? Because and there's a whole sub-consortium that deals with that question, because originally you would think it's a genetic question, right? So someone might be genetically African, for example, or European ancestry white. But that is actually very complex to assess because, and again, we could stop this lecture and have the rest of the 30 minutes discussing about whether or not what I'm going to say next is true, is that you usually define it based on those self-identified questionnaires, right? And for example, Hispanic is a question mark, is a question on the questionnaire, which could include people who are ancestry-wise could be European white or African black, right? And then the idea is that it's culture and lifestyle what makes the ethno-racial subgroup. So that being said, is that there's actually studies that show if you go by pure genetic ancestry, right? So if you really look at sort of the genotypic profile in terms of their lineage, that actually often doesn't give very interesting results, and you have to use these more culturally self-identified features. I know a lot of question marks on that. So we had to, in this study, because frankly, it's also not our expertise, we had to follow the guidelines by an NIA subgroup that works on this stuff and told us that's how you're supposed to do it. That's how we did it in the other papers. But sort of the mixture of the, let's say, cultural definition of the ethno-racial group and the genetic one is a very, very complex topic. And in any presentation ever, you run into some kind of resistance for whichever stance you have on that. Yeah. Rightfully so, it's complicated, yeah. Okay, peripheral. So now this is blood, and here we had one very specific study. This is in the ATNI, and so if we jumped on the very popular train these days of biological aging clocks, I have my own specific opinions on it. So the fast version is that I believe if these clocks put in, like, it should be called metabolic health index. That's what they should really be called. So your metabolites look older than they should be. I think the problem in the aging field today is that's a lot of consumer products that are now coming out that you're trying to sell you how old you look in terms of your blood profile, and science gets a little pseudoscience-y there. But sort of what we try to do here is in the ATNI cohort to look at acylcarnitines. It's a species of metabolites that I'm sure people know. And the fascinating thing about acylcarnitines is that we know from many years of research that the blood levels of them reflect certain mitochondrial activity. Especially, and this, again, if you think about a couple membranes away from the mitochondria in blood, but they're represented well. This has been shown by GWAS. This has been used for decades in newborn screenings. So the blood stick, the heel stick, or the peripheral blood on the dried blood spots for newborn screenings. They look at acyl-kinetines for mitochondrial disorders. So the role of them as a mitochondrial readout in the blood is actually pretty well established. And we are working in fasting samples, so probably it's not mainly driven by glucose, but by fatty acid and protein catabolism. And so what we built, and again, this, bear with me to explain this. So the starting point is not the clocks yet, is we cluster patients by their acyl-carnitine profile. So what you see here, the background is just a hierarchical clustering, but only using the acyl-carnitines. A method has been out for many, many years, right? You know those trees. So these are all down here, all the patients or study participants, and you cluster them by similarity, and but in this case only on the acyl-carnitines, not on all of the metabolites, right? And now the text that you see on these is whether or not at that particular split, so for example, this split splits all people into sort of two halves, the left half and the right half, which of the Alzheimer's-related phenotypes are significantly different left to right. So you have tau, FDG PET, and cognition. So this is tau, this is glucose metabolism in the brain and cognition. So what I can tell you is the left side is much healthier than the right side with respect to those measures. And then if you go into the left side, there's actually another split. By age, there's an older subgroup here on the right. You can't see that they're older, I'm just telling you that. And they actually have very beneficial pathology. So they're old, but somehow really healthy. And so this toolbox, we called it SGI, which is it just stands for subgroup identification. It's sort of a fun toolbox. You can just put your omics data in that you want your patients clustered by and throw any set of phenotypes on it, and it generates you these trees that show sort of how the stratification of the patients according to the omics marker correlates with the pathology. And what was also interesting here is this, in ADNI, only has the Alzheimer's signal with the acyl-carnitines. If you cluster by anything else, the amino acids, the phospholipids, this was the Biocrates panel, then you do not see the main stratification of people by Alzheimer's. It's by like cholesterol levels or sometimes by sex and BMI, but only the acyl-carnitines have this association with with Alzheimer's outcomes. And now comes the biological age measure. I'm skipping the statistics a little bit here, but basically what you can do is you can fit a model that gives me, in addition to the chronological age, the time since birth, a acyl-carnitine-derived biological age. So they might be 70, but the acyl-carnitines look like that of an average 60-year-old. Or they might be 30 years old, but the acyl-carnitine profiles look like that of an average 70-year-old. Right? So it's something you can, with some error, statistically assess. And now, I'm just going to walk you through this. So if you remember, I said the left side are healthier. So cluster 2 is healthier than cluster 3. And you can see that while they're almost similar in chronological age, almost identical, maybe the right side is a little older, the right side is significantly older in terms of their bioenergetic age. And then if you go further down here, I told you this is an older group that are much healthier. You can indeed see they're older, but bioenergetically they're actually a little younger than their other group. So it seems like this measure correlates with how sick they are in terms of Alzheimer's. And in fact, it doesn't just work in terms of association, it also works prospectively. So if you go into these people and you split them into the ones who are older than they should be and ones who are younger than they should be, you see that over the next 5 years, those who are older than they should be actually decline faster in terms of their cognitive decline. They all decline, they have Alzheimer's disease, right? So none of these will be saved yet, but they decline faster. And that also works in other studies. We tested it in ROSMAP and also in the AGES study. And to add one more component to it, if you add a very specific, sorry, this is in the ROSMAP, I said it wrong. Age thing works in ADNI and the AGES. And then there's one specific SNP also related to this type. It's 2 SNPs that you combine into a specific genotype. And again, if you have the wrong genotype, you decline much faster than if you have the beneficial genotype. And putting it both together, it turns out that the only protected group, those that decline faster, they have the beneficial genotype and they have young aging at the same time. So they seem to have some kind of advantage. And in this paper, we didn't get further than speculating, but what's interesting is if you compare this with, so the effect sizes, if I were able to take someone with a good genotype and bring them over to the younger metabolic age by some kind of intervention therapy that we don't know yet, if we knew that, this would be higher than Nature Communications. That would be sort of the revelation. But if we were able to transition someone over into the younger age group, the effect size of benefit is about as high as that of the accepted FDA drugs that are currently being in use. So if we can transition someone over, I don't know yet. I invite everyone to try. So the candidates for transitioning over are surprisingly boring. It's because those are the mitochondrial health interventions that are already out there. So maybe it's nothing new and just need to do the standard stuff. But we believe that if it were possible to move someone over, they should have
nobody doing these trials?
Yeah, so we've tried to tag on, we have some keto data in a minute. And we
also physical exercise and stuff. People
Yeah.
say so for Alzheimer's all the time.
Yeah. So we tried as a bit of a, it's not an official project yet, but we tried where we have acylcarnitines to recalculate the age. And it does go down on keto. It does go up in spaceflight. So it seems like when you get shot up with a rocket, that seems pretty stressful. And some other ones. So we have we're trying to look into that on the data sets we already have. It would be nice to make a new intervention trial on this. But yes, some of these have acylcarnitine data. Yeah.
Then how is the peripheral acylcarnitine finding translate to the brain one?
Yeah, that's a great question. That was also that was one of the main reviewer questions in this paper. So what I know is from our data is that the brain acylcarnitines are not as affected in Alzheimer's disease, which of course begs the question, what is the peripheral one even showing here, right? So it seems probably more, and this is the big complication of Alzheimer's disease, you of course have the pathology in the brain, but the disease risk seems to be very much related to non-brain health, right? So people who are healthy, athletic, good exercise, good diet, seem to have lower incidence of the disease itself. Now, what that causal connection is very much a debate in the field. Why does your brain, with all the blood-brain barrier and so on, start to go wrong if you're generally not healthy? I think that is unclear. But this signal that we found, great question, is purely peripheral. It's not a brain signal. Yeah.
Do you have access to any B-vitamin data in this cohort? So essentially metabolic cofactors that would drive mitochondrial energetics?
We do. It is, at least in the postmortem samples, some of the metabolites of that pathway are downregulated. That's what I know. We haven't looked into it any further than that. You would think that could be a causally related factor.
And if you gave people vitamin supplements to drive improvements in mitochondrial energetics, it might help this data. Be interesting to try.
An intervention would be interesting. Yeah, no, it's actually a good idea. You mean as a supplementation? Yeah, it's interesting. Mm-hmm.
2 questions from the chat. Does the bio-age based on the metabolite correlate with the bio-age that can be estimated based on the body scans, like the muscle mass versus fat?
So I don't know for this particular so there's other bio-age measures out there, many, right? Transcriptomic, epigenetic, I think that's the original one, and then you can do sort of a DEXA scan and can get bio-age. So we know that they all correlate to some degree, but also show different components. Ours is this very, very specific sub-pathway. So I would predict it only partially correlates with the others, but I don't know that because we didn't have these body, the body muscle mass measures and so on for this particular cohort. So this is a sort of there's this world of bioage, and we have this side method. So I don't know the answer to the question, but I would predict it's not fully the same. And then I think the other question here is the Harvard, the methylation one. I see it on, it's the same answer. So they all have been described to correlate with each other somewhat, but we have made the specific one on only like 10, 12 metabolites. So I don't know the answer to the question. I think to some degree, if they have a general health predictive value, they should all correlate somewhat, but I'm pretty sure they will also have their own component that they see. Yeah. All right. Because of the nice discussion, thank you everyone. I'm gonna probably skip one of the sessions, one of the sections. So I'm going to just show one part of the nutrition because I really want to talk about the final project as well. So we had a keto diet, right? So everyone talks about it, everyone wants to do it. I wouldn't do it. But there's also now first indications that maybe long-term keto is not the best. I believe the only approved indication for it is some forms of epilepsy. There's anecdotal evidence, very strong anecdotal evidence from cancer therapy that, for example, metastatic cancer grows slower for people on keto. That's a very valid reason. But of course, sort of the bigger idea is it a good way to eat? And then there was this BEAM study by Suzanne Kraft, who and so Rob Knight was on the microbiome part of this here as a collaborator, whether it is beneficial in Alzheimer's disease and memory. This whole idea, it gives you some kind of healthy metabolic reprogramming and then you'll do better. I'm not going to answer that question today because this was a very small trial and not this study. So the study itself was trying to see if there's a memory outcome effect after a couple of weeks, and it wasn't a strong effect because it's just a couple of weeks, right, of being on this diet. But what we use this data for, instead of looking at if it improves memory, is what does the metabolic footprint of a keto diet look like in the CSF, so the cerebrospinal fluid. It's, if you think, it's actually behind the brain, so it's what the brain swims in. So if you have the blood-brain barrier is the blood, then the brain, and then it swims in the brain. So not physically behind the brain, but sort of in the cascade from the blood. And then we had got microbiome data here. And it's going to be a quick overview of what we found. So this was the Nightingale panel, and that's going to come up in a minute. So that's an NMR-based, well, you could call it metabolomics. I think Nightingale themselves doesn't even call it metabolomics anymore. They call it clinical biomarkers. So you get only 250 markers, and they're from a very specific subset. So you get the amino acids, some inflammation molecules, sugar, fatty acids, and then all kinds of lipoprotein particles. So it's a bit of a, if you're used to more sort of the Boehringer map style metabolic pathway coverage, that's not what this gives you. It's a very specific subset, but it's a very cheap and fast technology. So what happens when in the blood when they go on keto is, first of all the ketone bodies go up. That was a positive control. If they don't go up, you probably either did a wrong study or bad adherence or your NMR machine didn't work. And then you see all kinds of changes, and I'm just going to highlight a handful. So we see the branched chain amino acids going up, which is, and sorry, I'm simplifying a bit, generally considered good for you. We see, for example, citrate going up and other things. So the general pattern that we see is, this is purely by association, not causal yet, is that things that are known to go down in Alzheimer's go up in keto, and if they go up in Alzheimer's, they go down in keto. Of course, sparking some hope of maybe down the line, not yet, metabolic reprogramming that could be beneficial for Alzheimer's disease. Again, so far, this is just observation and conjecture. We don't know that yet, but at least in the contrasting it with Alzheimer's signals, it looks to go in the, so to speak, good direction. And then one specific side analysis we did with Rob Knight, again, unfortunately not causal proof, but maybe compelling evidence, is that, so the question was, why do the branched chain amino acids go up? And what we found under keto is a significant increase of branched chain amino acid producing bugs in the gut microbiome. Of course, we did not see physically, we didn't trace the branched chain amino acids coming out of the bacteria and go into the bloodstream, so it's not necessarily proof that that's what happened, but there's a compelling correlation that we see an uptick in, or an increase in branching amino acids and an uptick in the bacteria that produce that stuff. With that, I'm actually gonna, for the sake of time, because I wanted to leave the last 10 minutes for discussion, gonna skip the cancer, skip the dog cancer also. We also have some spatial metabolomics coming up now, but it's all in very early phase. And wanted to talk a little bit about our current, I would say, the hottest project, and that's a measurement in the Kaiser Permanente cohort. So especially here in California, people probably know Kaiser. We in New York don't know that so much. I mean, Virginia has Kaiser, but not New York. And the interesting research aspect of Kaiser, if you've never thought about it, is that they are both the insurance provider and the medical provider, right? So they have what many want is they have both the electronic medical records, everything in one system, and then that across all doctors, right? So if you're a Kaiser member, you go to Kaiser doctor, so your primary care will be there, any specialty care, everything will be at Kaiser. So theoretically, you have a full image of these patients' trajectories without having to find them from multiple systems or even files that are submitted. So their research bank is 400,000. They're all genotyped. And now what we did with them is we measured in collaboration, so it was this three-way Cornell Nightingale, which is that NMR platform panel, and our cell and Kaiser, we measured 55,000 for this NMR. And this is still unpublished, so some of the stuff I'm going to show you will look very rough because some people, I told that yesterday, I was still torturing my PhD student on Slack to send me plots for the talk today. So some of it is very, very fresh, so bear with me. But I'm just going to show you sort of a little bit of an overview of how we're thinking about this cohort. So first of all, the general research plan is that we have the metabolomics, we do have the genotyping data, which we're going to use mainly in the way of polygenic risk scores. And then we have electronic medical records, which is really vast. So what's important to understand is it doesn't contain, people thinking AI and language models, it's not the written notes. There's all kinds of AI governance issues still that you can access them. So it's mostly what's in the structured data, diagnoses, medication dispense. Well, technically, yeah, we know that they received the medicine. We have to assume they took it. And then, medication diagnosis dates, visits, and all of those features, right? And it's not even us, so there's a whole dedicated informatics team that we work with at Kaiser that do the data pulls for us, so we don't have to deal with that. So we tell them, hey, we would like to have all ICD-10 codes so-and-so for diabetes and complications, and then they pull that for us. And what we would like to figure out is if the addition of the NMR sample can help in predicting disease incidence, sort of risk of the next few years, and within the sick, if it can predict complications. For example, in diabetes, you get diabetic retinopathy and other things. And the earlier, with the higher accuracy we can predict it, the more you could intervene. So what makes the NMR platform interesting for this is that it's absolutely quantified, and there's a lot of samples out there, including, for example, the UK Biobank is fully measured on that platform, so you can always have some reference to compare it to. And they're like $20 a sample. So there's a reasonable idea of making this clinical. Still a way to go, but in Finland, in fact, they are using this platform in actual clinical rollout. Not all the experimental biomarkers, but for cholesterol measurements, they're already using this in the real world. So now I'm going to first show you a little bit about what kind of data you have to deal with. So first of all, you have all kinds of metadata complications. So this is longitudinal data. This is a person with, and I'm showing you the worst case examples here. Not everything looks like that. For example, you have BMI measurements here, and then the red dotted line for these individuals is always when they got the blood draw from Kaiser, so when they entered the study. And then everything in the plot is their BMI measurements. So you can see for someone like this, well, we didn't have a BMI measurement, which I never understand why not, just height and weight and put it in the thing. But sort of on the day they gave their blood sample, there was no BMI measurement, right? But as you can imagine, you could reasonably assume where their BMI was giving this curve, right? For someone like this, it looks very different. So we don't, it's very hard. What do you do, right? Was this a data error? Did they just lose a lot of weight? They probably did. There's actually a lot of heavy weight loss in the early days of COVID, which is exactly when this was. But you have to start to make some assumptions already on the metadata. You probably have to, if you need BMI for your models, you probably have to give them the next closest BMI value somewhere here, but you need to remember you made that assumption, right? And then what are you supposed to do with someone like that, right? So you're supposed to say they don't have BMI, or also they went apparently from underweight to obese. So there were some decisions that had to be made, and we went back and forth. Usually we just take the closest point because the best we can do. But that's what you have to deal with. And then, for example, this was medication dispense data. So when they picked up what kind of medication, so that's what we use to judge what the medication they're on. Why I'm showing this is that this is a statin, it's actually not a calcium channel blocker. So there's also some metadata problems, some misannotations. So all of that took us almost a year to go through and sort of clean up the metadata set as much as possible. It's just the stuff you have to also do in addition to the fun statistical analysis. Then we have technical issues. So they drift because sometimes it takes a few days from needle to freezer, actually sometimes scary long, especially for metabolomics purposes. This is phenylalanine, it drifts up over time, and at least we know the time, so we know how long it was. So we can correct for it, and hoping then it stays stable enough, so you kind of correct the effect out. And then we have interesting, that's not a technical issue, it's more like a feature, is if you want to compare to the UK Biobank, for example, UK Biobank is plasma, and Kaiser has serum. Now, for some metabolites, that's fine, for others, it's not. And you see, I have some examples here, we talked about it yesterday. So for example, glycine has a multiple standard deviation difference between Kaiser and UK Biobank. So this makes this difficult to compare. And of course, what you don't know is it because of the plasma serum biospecimen difference, or is it actually a cohort difference, right? Americans versus the British. So we had to make individual decisions. For example, this one we're correcting out. So we were just putting the two peaks, the two distribution peaks onto each other because we decided, no, the Americans do not have, in this case, KPRB, so the Americans do not have multiple standard deviations, higher glycine levels, just not what it is. This is a matrix effect of the specimen. But another thing you have to sort of overcome and work your way through. And all of these are assumptions we now have to defend into the future. But now I'm going to show you some results. So how this plot works is, so we have three basic panels. So base is just predicting by age and sex, that's it. Clin is sort of we call it sort of if the doctor sits in front of you and has no further lab measurements, what would they reasonably use? That's age, sex, BMI, and blood pressure. And then marker means that for whatever disease we're investigating, the number one biomarker that's being used in the field. So for diabetes, that would be HbA1c. So those are the red dots. This is the predictive power in terms of C-index, so survival, time to event, into the future prediction, AUC. And then the blue ones are when you add NMR measurements on top into the prediction model. So spend $20, forget about the fact that we still need regulatory approval, and that's the benefit you could get, right? And it's actually surprisingly high. So you get good jumps in terms of predictive power from all of the models, including this one, because that's the one that includes the A1C, the measure that people use for diabetes diagnosis, but also prediction in the future. So across the board, the metabolomic panel gives a good boost. And that's the case also for other diseases. So we look at fatty liver disease, where the biomarker is ALT. We look at chronic kidney disease, where the biomarker is creatinine. And we look at heart disease, where the biomarker is LDL. Sometimes the improving effects are moderate. Sometimes they are, here's even a little bit down, which is probably a stability issue. But across the board, it seems to be worth it to add the NMR to these samples to increase the predictive power.
within the NMR, what are the features that make it go? Is it the lipoprotein, the cholesterols,
Yeah.
amino acids? What is it?
Yeah, I can't tell you fully yet because this is the result from yesterday. So we're still dissecting the models. But I do know. Well, we've done a few iterations of this. So for diabetes, for example, it includes also it can include the blood sugar. So there you would be like, okay, that's not a special finding. But apparently, then the blood sugar combined with the A1C, which is sort of one is the short-term measure and one is the long-term measure, helps. In the heart, it tends to be the different fractions of subparticles of the lipoproteins. Now, which ones exactly and why that one and not the others, that I cannot answer yet. We have to dissect that. The feature importance of these models, we don't fully know that yet.
Have you looked at the models with biomarkers alone without age, sex, BMI, blood pressure?
Good question. No, we have not. We should look at that. Yeah, we have not. So that would be another. It would just be the biomarker basically. Yeah, that's a great idea.
Or looking at covariance between the SAGE panel,
Yeah.
or the Nightingale panel, and
Yeah.
the key markers.
Taking a note here. That's a good idea. Yeah, and then which features? Yeah. So I'm taking live notes. That's where you answer the questions in the and so one so first of all, for the statistically inclined, there was a very specific sub problem here, which is that it's actually not statistics, it's actually very medical, but meets statistics, is a good subpopulation that have no lab values, except the NMR. They just came in, probably they got an email, hey, would you want to come to Kaiser? Enroll in the court, they went, gave their blood sample, didn't even get a regular checkup at the doctors, they just donated the sample. And so and we can't just completely ignore them because these tend to be quite healthy, which makes sense. Someone who doesn't even go to the doctor, right, doesn't even get a regular checkup, usually was either someone who doesn't take care of their health or tends to be very healthy. And we can show that. I don't have the curves here, but they're all healthier than the rest. So we can't just, in our statistical models, missing values are not something that is very good to use. So you can't just completely ignore them because what you start doing is you accidentally do your model in this slightly sicker subpopulation, ignoring those 10,000 healthies who have never received a value. And we had a long time working through statistical models. So basically what you're doing is you're presenting to the model the biomarker if it's available. And if it's not available, you're at least telling the model that there was no marker. And that then captures the statistics. You can model all 55,000 of them, including the 10,000 that are missing. And the model will use kind of what a doctor would use. They would say, if you have an A1C value, I will use it. If you never got one, you're probably on the healthier side. And the model, we don't tell the model that, it's going to find that out. So that was an interesting statistical detour, heavily involved with the medical background of these people where we had to deal with missing values. This is something you don't get in the UK Biobank, because UK Biobank is a perfect population cohort. Everyone got an A1C value. That's sort of the problem you get in the real world cohort. I know I'm running out of time. I'm going to give the last.
Did you use metabolic profile to predict AD?
In this study, AD, no. So the Kaiser, if that was the question, the Kaiser cohort, because they are a little on the younger side, is not very good in capturing neurodegenerative disease. There's not many Alzheimer's patients in here, or Parkinson's patients for that matter. So this project is really more about these classical metabolic diseases. We also have a bunch more heart disease, subvariation, stroke, and AFib and so on, which I'm not showing here, but it's more that, it's not the Alzheimer's and Parkinson's. And maybe the last finding, because that was a shocker for us, is that at least to our results, if you add the polygenic risk scores into this, it does not help beyond already the NMR. So it adds value. So if you have just a regular model and then you add a polygenic risk score, and we took sort of known polygenic risk scores, and then the Kaiser people calculated the score, so we didn't even do that, but we just took the score. But what you really want to compare is the blue dot and the green dot, and maybe there's a tiny little addition of the genetic risk, but it's not much. And this goes across all models. So if you always compare this blue and the green dot, there's just a little bit. And sort of the argument that we're trying to make in this paper is getting a $20 blood test, yes, getting a genotype profile is sort of a much bigger clinical ask, right? And it seems as though it's also not necessary. This was sort of a bit of a shocker to our genetic risk score friends in the project here. And I know I'm already running later than I wanted to because I wanted to leave a few minutes, so I'm just going to close it here. Thanks for listening, and are there any remaining questions?
So, I was wondering, for the plasma versus serum, is there any data set that has both of them on the same sample, so you can build a model that converts one to the other?
Yeah, I believe so. I don't know, Oliver, if you know any of the. I remember one small data set that had about 50 or 100 plasma and serum samples, not on this platform though. So that would be the gold standard where you would do that. We had another one where there's a Finnish cohort that has serum. So it's now we could take 2 European cohorts at least to get sort of that transatlantic factor out. But what you're describing, that would be the gold standard. Yeah.
I have a data set that you can play with.
Oh, really? Yeah, that would be because what we really need to do is do it in an informed way, not with 20 assumptions. That would be the better way. Yeah. Shane?
I'm just repeating the chat. Laura is saying there is a Kaiser Alzheimer AD cohort.
Ah, the Kaiser, yeah, they have a subcohort. That's right. So we're talking to the good point. We have not talked about that yet with the Kaiser folks, but yes, you're right. That's a good point. I've heard that there's a subcohort. Yeah.
Yes, Laura also posted the PI's name for you to reach out to. And back to the new Alzheimer or nutrition. How does the GLP-1 usage might compare to the ketogenic diet impacting the?
Yeah, I wish I knew. GLP-1, of course, I mean, they haven't been around for so long, but now the studies are coming out. What we're interested in, and we just haven't been able yet to find the correct data for that, is to do acylcarnitine profiling in GLP-1 intervention, because that's something where we could certainly check if the bioenergetic age goes down, which would be the assumption. But yeah, no, great idea. Definitely on our radar. Haven't been done yet. I see there's a second question. How stable is the NMR signal over time during acute illness? Yeah, so I think when you say acute illness, do you mean critical illness or you mean during infection? Yeah.
Sepsis.
I don't know. So because the Kaiser, I mean, technically speaking, if a Kaiser patient went to the to the emergency room, let's say, or the hospital, we would have that data in there as well, but we usually exclude those encounters. So the, sepsis. We had a totally different study in sepsis, COVID sepsis with metabolomics, and there's massive changes during sepsis that are probably not anymore just physiological biomarkers, but just escalation of the system. For the NMR, I don't know. I don't know if anyone has done it in that setting. For the acylcarnitine. Sorry, I'm monitoring the chat. So acylcarnitines in sepsis would probably be an interesting thing to look at, confounding, but it's not the type of setting that we usually work on. We're more like sort of in the chronic disease business, except for the COVID detour that I think all of us did. I'm just going to read off another question. Is your interpretation that polygenic risk scores does not add because metabolic data captures the underlying genetic data? Yes, so that's what my interpretation would be. Whatever the genetic signal wants to add to predictive power is also contained in some complex signal propagation way in the NMR data. At least that's what is suggested by our results. Doesn't mean you don't need genotypes, but at least for this particular piece of the risk, you don't need genotypes.
I also think it is more metabolic disorders rather than like inborn errors.
Right, yes, exactly, yeah.
So about the genetic analysis.
Yes.
So first, PRS will only give you the very, very extreme cases.
Yeah.
So the distribution might not seem shifted, but if you look at very top 1%,
Yeah.
I think you will see a difference. So that will be the application. Second, PRS will not have rare variants, which is probably a big chunk of your variabilities.
Yes. I think so. I agree with that, and so I think maybe I did what I meant to. So at first I wrote genetics doesn't add any predictive power, and then I wrote PRS doesn't add. So that's maybe the more accurate. So the polygenic risk scores as they are currently calculated don't add. Whether genetics couldn't help here at all, especially with rare variants, that's a totally different question.
Also here, I think if you look at the very
The top
extreme
ones.
scores,
That's a good point.
it might act.
Yeah, no, actually, I'm already that's very good feedback that we're going to look at that about the top ones. Yeah.
And feedback?
It's interesting that you saw the osmoregulation feature pop up in the Alzheimer's disease program. I was wondering, I noticed those are all compatible osmolytes that you showed there.
Those were all the ones we could see in the panel, yeah.
I'm wondering whether they are somehow related to not osmoregulation, but things that are like, for example, lithium is a drug that's used for treating schizophrenia, which basically acts on inositol. But there's some connection there with the other cerebral diseases that somehow lead to regulation of these compatible osmolytes.
Interesting. Okay. So it could be so it's not actually osmosis per se.
And not osmotic pressure related, but somehow related to these compatible osmolytes that have some sort of protective function. Because these are also called compatible osmolytes, are sometimes also called micromolecular chaperones, as opposed to macromolecular chaperones in each of proteins. So micromolecular chaperones protect like macromolecules in terms of sort of reforming protein structure and so on and so forth. So I'm wondering whether there's like a function associated with that aspect rather than like osmotic
Pressure. Yeah, that's a great question. It sounds much more appealing how you phrase it than thinking it's all about just pressure. I don't know the answer, but that might as well be it. Very interesting point. Yeah.
Edwin?
Yeah, I've been thinking about the metabolic reprogramming for Alzheimer's. I think conceptually, in order for that to work, there would need to be a different way that diseased neurons like take in metabolites. Are there any targets that you suspect would be disproportionately brought in to a diseased neuron compared to a healthy one?
Yeah, that's a good question. So I think so basically you're asking, if I understand the question correctly, if there is metabolic dysregulation observed in the sick brain, how did that happen, basically, right? Is that what you're asking? How did it change?
Yeah, and I think if you observe metabolic differences in Alzheimer's, if that's all like output,
Yeah.
then reprogramming wouldn't really do much because it's not interacting with the disease itself. So I guess I'm asking, what's the intake that could be acted on?
Yeah, so it's a great question. And the problem will be that whatever you're observing is a systemic effect, right? So you have changes already in the periphery before disease and during disease, right? And then you have this complex issue of the blood-brain barrier, but it does let a good amount of substances through. So now you're already flooding the brain with things from the outside, right? So that will affect the equation you're talking about. Then come the neurons, then come what they produce, and then come what's potentially being transported back, right? So it's, for example, we have one finding where in the CSF something goes down, but it doesn't necessarily mean something is downregulated. There's actually certain evidence that it goes into the brain, so you don't see it anymore in the CSF, right? So, but what you unfortunately have to do is you have to make speculations on flux based on the steady states that you measure outside or in the different compartments, which can be very hard.
It would be kind of a difficult thing to try and isolate.
Yes. So I think you're asking the exact right question. It's just very difficult to answer from these snapshot data sets that you would have to trace the molecules actually to see that. Yeah.
All right.
Yes. Laura commented on the PRS score. Maybe the younger the cohort metabolic data is collected, the more PRS predicts and less metabolic data predicts. Can you subset the data so you can look at the prediction for those who had sample at younger age that had been in cohort long enough to have developed certain disease and you were
Right.
working with?
I get the question. So basically the idea being if you're younger, you haven't had yet time to accumulate all these metabolic problems, but you are already walking around with the genetic problems, right? It's a good question. So the thing is that usually we're predicting disease here, diabetes, in a 5-year horizon, not much longer. So the young already don't have much disease is the problem. Like I like the idea of the analysis. Is there a difference in predictive power in the young, metabolic? And I think we should check that. But I think it might suffer from the problem that the young already don't have a lot of disease, at least in this cohort at all. So that might be an issue. But I like the idea. We should definitely check if there's a difference in the very high percentiles, and also maybe the young versus the old. Yeah.
One more comment. If you put it in a summary take-home message, how would you conclude?
All of it in a take-home message? Well, I don't know. Metabolomics is alive and well, but no, I think I would say okay, maybe as a take-home message from me is that really we are now at the stage where some of it is borderline crossing the clinics, and in some countries has. It's not the mass spec version, but the NMR version, but it's sort of getting in there, while at the same time you can still do this very basic, I haven't even talked about the cancer stuff, a very mechanistic research. So I think it's now trying to really span the spectrum all the way into the clinics, which I find very exciting personally.
All right, in the interest of time, I thank very much the speaker. Thanks so much for a lively discussion, and also both online and here in the room. Thank you so much.
Thank you.